While you guys do that, I'm going to introduce our next speaker, Dr. Andrew Walker. He is one of the most influential viticulturalists of modern winemaking. Dr. Walker is a pioneer grapevine geneticist. He's the Louise Rossi Endowed Chair in Viticulture and Enology and began breeding grapes at UC Davis in 1989. His research is focused on developing new rootstocks, which are resistant to a host of different pathogens and vectors, and teaches two parts of the three-quarter course on viticultural practices, and is also serving as the chair of the Horticulture and Agronomy Graduate Group. So I'd like everyone to welcome Dr. Walker. We're really honored to have him here. And without any further ado, Dr. Walker, if you would take it away.
We'll see. There we go. Is that okay?
Looks great.
Okay. Good morning, and thanks, Daniel, for having me by the chat. And Greg, thanks for your answer to that last question. It summarizes about half of my talk, so that's an effective way to get it going. And it's part of the whole problem, right? How do we address a changing climate, and how rapidly will it be changing, and can we actually make convenient enough switches in our agriculture and our practices and in our variety use as well? And that's more or less what I'll be discussing today. So how do you adapt to warm weather? You move. That's the easiest. Well, no, not really, right? And in fact, worldwide, we've invested billions of dollars, trillions perhaps even, in marketing names and places, and emphasizing terroir and how important a site is over other issues in terms of viticulture. So that's really probably not likely to happen. So we're going to have to adapt the varieties to some extent as well. How can we do that? Well, there's all sorts of work that's gone on shading covers and misting, and that'll continue, and it has some impact, fairly large impact sometimes, but there's always secondary problems. So it's a means of addressing the problem, but maybe not a practical one. We can alter phenology to some extent with pruning and training, and we can double and triple prune, and they already do this in parts of the world that are more tropical, and you have a 3 or 4 month period where it's fairly dry. You can prune to address that cycle of growth. So you can switch the whole phenology of the vine in many ways, and you can crop 2 or 3 times for that season and get rid of 2 of those crops. So it depends on the value of that crop again too. But in terms of fitting the window where it's not going to rot and decay from fungal issues, it's possible to actually adjust growth quite a bit. We can select new clones. We'll talk more about this later, and it's probably the least effective way of going about doing it. And we can just get used to the idea and get back to breeding grapes again. And of course, it's been a very long-term process over history. Remember when grapes were first discovered by humans? They were wild vines, and we still have wild vines growing all over the planet. There's many different species, somewhere between 20 and 30 and perhaps 60 to 100. They are male or female flowered vines in the wild. So they're either male flowered or female flowered. So they're obligate outcrossers. And in fact, the genetics of grapes are a lot like the genetics of humans. Grapes are massively migratory because of birds, and they're outcrossed, obligate outcrossers. In other words, they have to be a separate male and female plant, and there's a lot of mixture of gene forms, gene alleles in that process as well. So there's a cluster of American species, not many in South America, which is quite interesting, given the tropical jungle aspect of the Amazon and everything else. There's really not many grape species in the Americas. In North America, Central America, plenty of species. There's a cluster of Asian species we've become much more aware of over the last years, and they're quite remarkable in terms of characteristics. They have tremendous fungal resistance. They have tremendous cold tolerance. Some actually don't taste bad, so they're fairly easy to hybridize with Vitis vinifera, with the cultivated grape forms, to mix in new characteristics that way too. Within the Fertile Crescent is where Vitis vinifera evolved, we think, which is not really true, since the birds had already spread it all over the Mediterranean basin. And in fact, the grape varieties are more or less characterized and grouped by location, and those locations were determined originally by birds and later by people when they first started invading and crawling out of Africa and moving into the Mediterranean basin. They moved grapes at the same time too, and they started selecting grapes as well. So how do you tell what these things look like? You can actually more or less tell what you're looking at by appearance. The Central Asian grapes have very large floppy leaves. These are the table grape types, and big irregular teeth, larger, more oval berries. The Eastern European types sort of move towards that table grape form to some extent, but they're smaller leafed, smaller berried. They have sharper teeth, oftentimes a bit more typical. And a good example of an Eastern European grape would be Zinfandel, and we'll talk more about Zinfandel in a while. It's a nice poster child for a lot of genetic issues. The North African materials bridged the East and West very effectively. There was a lot of migratory movement in that direction, more so than in the northern part of the Mediterranean, probably. And grape types moved in that process well before formalized agriculture and formalized banking systems. Back when people were still hunter-gatherers, they were moving grapes back and forth across these corridors too. They discovered things that fruited better. They moved them and vegetatively propagated them, and that became part of the process. And Western European ones are the ones we think of the most. They're the 20 that Greg was talking about, more or less. There are very few outside that realm. They're mostly moderate to cool climate varieties, in fact, and not necessarily very well adapted at all to hotter conditions. So that's the real pressure on getting us to change and adapt and perhaps move forwards more rapidly. So the birds move things, hunter-gatherer societies move things. We had primitive agriculturists that started popping up and recognizing how plants had benefits. And of course, we were selecting for all sorts of characteristics across the planet at a very, very long time ago, much further than we think of in terms of the rise of agriculture. At 12,000 years or 11,000 years ago, it happened probably instantaneously as people started looking at these plants and thinking about how they could move them and adapt them and which ones fruited better. So there was grape breeding going on as well for quite some time. There's no evidence of it, so I can't be challenged from that statement, but I think the evidence is the amount of variety we have and the amount of very regional specific characteristics we have as well. So again, the wild vines are male or female. This is a picture of a hermaphroditic flower here to the side, a perfect flower that has both pistil, the female parts, and stamens, the male parts. And in fact, the cap, which is that little petal unit that we call the calyptra or the cap, pops off and it splits open from the bottom, not from the top. So most flowers open from the top and expose the pistil to pollen. And in this case, we have them opening from the bottom, and immediately upon popping off, pollen is shed from the stamens and it self-pollinates these flowers. So there's almost no way to make a cross unless you remove the stamens. So that's the very interesting thought. I'll let that linger for a while and think about how that works. The cultivated grapes are all self-pollinated, except for a few, and those few are females. There are various varieties that are around the planet, not many, a few handfuls of female flower varieties. They're very valuable for breeders because a breeder, if I want to make a cross, I've got to isolate the maternal parent from the paternal parent. The paternal parent's fairly easy, that's just the pollen source. But the maternal parent, if it's perfect flowered, I have to remove all those stamens before I can make that cross. So they have to be plucked off individually with a forceps. It's not pleasant work. It's always hot. It's always windy. But it's a process that has to be done if you're going to make a cross. Here's a picture of some female flowered grapes on the far left-hand side, and male flowered grapes across that side on the right. And the males, all you can see is pollen. It's just a solid mass of anthers. And on a female grape, you have much, much smaller clusters, far fewer flowers that form, and they're all pistils. You can see if you look very closely in that picture, you'll see that the stamens are recurved. They're bent backwards and non-functional. So again, we have to isolate that maternal parent, and we have to emasculate it before it blooms. So before the petals pop off, we have to actually grab the top of that united petal mass, twist it very gently to the side, and pull off that petal plus all the stamens at the same time. And we have to do that. It takes about 2 hours, 2.5 hours sometimes to do a larger cluster. In general, by the time you get halfway through, you throw the rest away because you're tired of plucking off those stamens. Probably the reason why I have trifocals now. It's not an easy process. If you don't do that, wind will carry pollen from other sources. Bees will move pollen from other sources. But if you can emasculate them, if you can effectively separate them into maternal parents and not self-pollinating parents, you can move pollen and make those crosses. Okay. We now know that most of our crosses were hybrids of two different grape varieties, and varieties oftentimes that are known. And there were centers of breeding that was for quite some time. And if you look back and think about the origin of these, what we think is the origin of these varieties, it's quite interesting because it really implies that people were making these crosses for a long time. That was first discovered when we were looking at different ways to identify clones and fingerprint varieties, and we came up with a simple sequence of repeat markers. This is Carol Meredith's work here in the department that was quite useful and beneficial for a lot of things. But it allowed, her goal was to go forwards and identify markers that could help distinguish clones. That was one of the major issues. We couldn't distinguish clones. You can't tell which was which, so bookkeeping becomes so important in the process of following these things. And it didn't really work very well. In fact, it became the new definition of a variety. It was all the variants that fit within an SSR profile. Things we thought were unique now were clonal because they fit within that profile as well. This process is a bit limited by the completeness of your variety database. And now there are well over 1,000 varieties now in the database. So if you find an unknown, you can match it with those types. But more importantly, as you go through and look at the database, you can actually identify parents of things you think are the final progeny. We can go back and recognize the parents. And the parents are hermaphroditic all the time. And why do I keep making that point? Because someone had to isolate the maternal parent, and they had to pull off all those stamens. So these hybrids that we think about as potentially being spontaneous probably were not at all spontaneous, but planned and directed by people in terms of isolating that maternal parent. And there's lots of examples. Cabernet Sauvignon was one of the first ones, cross between Cabernet Franc and Sauvignon Blanc. And the big Bordeaux groupings but that grouping was mostly Gouet by Pinot Noir. And we now know a tremendous number of these things. So it's really, to me, evidence that breeding was going on for a long time because they're all hermaphrodites, and the hermaphroditic types, there is some outcrossing and there's some pollen that blows in crosses as that occurs naturally and unnaturally or by a breeder's hand, but not many. And it's interesting that all these varieties trace back to something. That information was used to look at Zinfandel, and of course we knew it wasn't Californian. There are no vinifera grapes that are Californian, but we weren't sure where it was from. By appearance, it looks like it's from Eastern Europe, and you can look at that whole grouping of varieties, all the Hungarian materials and the Serbian and Croatian materials. They have a lot of similarities as we go through. So that work allowed us to know now that Zinfandel is really Croatian to start with. And maybe the Hungarians argue occasionally that it's Hungarian, but the Hungarian Empire, of course, was huge and controlled a lot of property and possessions and perhaps varieties as well. But we now know it's called Tribodrag, which is even a more difficult name than the last one that we thought it was. And we don't know both of its parents. And in fact, one of them may not exist. So that's one of the problems with going back to the very beginning to look at these things, is unless you have these materials in collections or you have the DNA profiles of them, it's very hard to make that final step sometimes in determining what parents were which. So how else can we adapt to warmer weather? There's a lot, again, the viticultural tricks we can play and practices we can do to develop things. There are some clonal differences. There's probably a week worth of week to 10 days plus occasionally difference in a lot of phenological cycles with the clonal types within a variety, but oftentimes they're not that great, and it's not that distinctive. We can do genetic modification, and that will probably come to that eventually, but at no time in the near future, apparently. Society is not really pushing forwards in there, but we now have the technology to alter pathways, to look at acidity differently, to look at color differently, to look at probably larger scale phenological and fine growth aspects as well, and modify those things internally. Or we can breed, and I'm the proponent of breeding to use these things and use the variation we have and move forwards and address some of these issues that way. So as Greg intimated, there is a very large and diverse germplasm pool, thousands of varieties, probably 3,000, perhaps more. At one point, people used to talk about 12,000 varieties and then 5,000 varieties, and now most of us agree that it's closer to 1,000 or to 3,000 perhaps. And a lot of them, of course, are synonyms, and it's very difficult to tell, again, because a lot of these materials, we've lost the ancestors of them, and we have the progeny of them, but it's hard to tell where things came from sometimes. But they do have a very wide spread, from the very hot North African deserts all the way through to the high altitude Alp areas where vinifera grapes grow. There's a lot of variation we can select from, tremendous variation. And classical breeding in general, in terms of making new forms and utilizing these diverse grapes as parents, has a lot of potential. And again, when we select in a grape breeding process, we're selecting for a phenotype, not for a single gene change or a locus manipulation. Genetic manipulation misses that aspect, and it misses the genome-wide interactions that the genes have as well. So the classical breeding, we're looking at a whole assemblage of traits all interacting with each other, and it's probably the best way to go about addressing these things. And it doesn't take very long. In fact, you can make a seed-to-seed generation in 2 years if you push the plants, use a few tricks, and select for that trait in general. So you can actually go through generations, multiple generations, very rapidly, and it's not as long and painful and arduous a procedure as thought. It is not inexpensive. It's not cheap. You need a lot of space. You need a lot of materials. And if you develop the genetic markers for traits you're trying to chase, it can go more rapidly. But again, it takes time and effort. What are the problems with classical breeding? Well, one of them is the lingering distrust from the phylloxera crisis. So there were 3 approaches to controlling phylloxera in Europe in the 1860s through 1890s or so. And the first one was to kill the phylloxera. That didn't work very well. The next one was to breed hybrid direct producers, which were hybrids between the American species with phylloxera resistance and vinifera itself. And they were looking for resistant roots and high quality fruit. They didn't find it. They didn't work at it long enough, but a lot of that material has tremendous cold tolerance, has tremendous fungal resistance, has a lot of potential in other areas, but we never went to that final stage. We didn't push it far enough. And meantime, the rootstock effort to solve phylloxera was sort of in the background at first, and later became the primary solution to the whole process, and allowed us to leave the varieties the same. Right. And so when we finished the phylloxera crisis, we had this huge mass, probably 1,000 or so of hybrid direct producers that we sometimes call the French hybrids. Those materials were very, very productive. That's what they were selected for first. Remember, there was a huge world looking for wine and there wasn't any. So we were left with higher producing varieties, and a lot of that material that was selected initially was really more like table grapes than wine grapes. We went through and looked at that. And in the end, those plants were sort of legislated out of existence as we went back to using pure vinifera. So the effort to really finish them off never really occurred 100 plus years ago at this point. We still got to that problem, is what do you call these things? And in fact, in Europe, we typically separate how we produce varieties in terms of breeding into crossings versus hybrids. And hybrids have an alternative other than vinifera species in the background. And the crossings are pure vinifera. They're all hybrids from a genetic perspective, but they were looking to distinguish pure vinifera from American species material. And again, what are you going to call it was always the big problem. How do you integrate it into the marketing process, and which ones would be important, which ones wouldn't? And again, clonal improvement really is a change within a variety, and it's a very incremental change, and it's not going to allow us to address the climate issues rapidly, or even the ones that have already occurred. We need to move ahead, and we need to move with larger steps, I think, overall, in terms of how we're going to address these issues. So potential of classical breeding has been amazing, and we discovered this with our Pierce disease breeding here, how easy it was to go over a very relatively short time period with multiple years of back crosses, and it was just pushing an aggressive selection that really moved us forwards most aggressively. And it was my technician, Alan Tencher, who was really instrumental in getting these things to the final stages now and pushing them through to releasable materials. And we went from the material that was essentially not edible, let alone fermentable, many years ago, that was processed with rupestris and arizonica, and we moved that into vinifera through 3 to 5 generations of back crossing. And we've in that time period lost the very peppery, herbaceous, spicy character of those original wild species. We've lost the blue-purple pigmentation, which we wanted to get rid of earlier on. We've lost the extremely high acidity, and they look and they produce like wine grapes. So it didn't take that long. In the past, the biggest mistake we made in breeding disease-resistant wine grapes and table grapes, for that matter, was accepting the fact or allowing ourselves to be detracted or distracted by the need to make wine at each generation, which is a waste of time. We know that the early generations of these crosses are going to have poor quality wine. So let's move forwards and cross them back to vinifera to improve the vinifera aspects that we were looking for most rapidly. And we did that. We de-emphasized winemaking till the very end. and then decided with enough material, here are the better quality materials within that, knowing that we would find them because it was almost impossible not to once they went to that higher percentage of vinifera. So we've done that, and we've done that with Pierce disease. We've done it with powdery mildew. Powdery mildew, we've been more concerned about coupling different genetic forms together, different genes together, so we'd have a broader base of resistance. There's evidence that that relationship between resistance and the fungus's aggressivity is very, very delicate, and we need to have more genes, not fewer genes, to address it. So we now have a lot of these little flagpoles here are representations of genetic markers and the chromosomes or the chromosomes and the genetic markers that exist on them. And we need those to be able to follow these different traits because, of course, the phenotype, what these look like, they're resistant or they're susceptible, is the same across these different resistance backgrounds most of the time. So we need the genetic markers to follow those through. So we'll look for the markers, and they can tell us, yes, we've combined this form and this form and this form together. Now we know they're working in concert, and we can challenge them more effectively that way. So that's been going on for a number of years here in the group as well. We've bred up now 5 PD-resistant wine grapes that are almost 98% vinifera. They taste like vinifera, that look like vinifera, they grow like vinifera, which is useful too. They've been released now, and they're being utilized in sites across the U.S. and California. We're in the later stages evaluating many more of these, and as I approach retirement, I'm thinking, well, we better speed it up here to get these down to a handful or two that are useful. And we're using the same sort of strategies with the powdery mildew resistance and combining that with PD resistance. So those things have all gone together. And really, if you think about it, it didn't take very long. And we know a lot more now than we did then. And if you can define a trait, particularly in terms of a biochemical approach, we can define it precisely, and we can trace it and track it that way and breed for them more effectively. So when we have these tools, we can develop genetic markers, and then markers will allow us then to look at seedlings that have just sprouted, see if they have that trait, throw them out if they don't, and maintain them if they do. And so you can really focus your energy on those situations. So far, it works most effectively when these traits are controlled by one gene, perhaps 2 on occasion. It's not as effective when they're controlled by many genes. And of course, unfortunately, a lot of these traits are controlled by many genes. They're not resistance traits, they're phenological or growth traits. And we're going to have to address those too more effectively. That'll be for the future too. I want to acknowledge my funding sources, particularly ETS Labs, who have been really, really generous in doing juice assays for us over the years. American Vineyard Foundation, the CDFA, PD Grist Board, which was just renewed again. Thanks everyone for voting for that. The Table Grape Commission, the Rootstock Commission that does our rootstock work as well, and the Rossi and Dow Chair and Viticulture funds that have really been beneficial in sleuthing out a lot of these wild species and where they came from and how we can utilize those too. And Ian J. Gallo looking at drought and salinity tolerance in some projects we worked with them years ago as well. And with that, I'll say thanks and take a few questions if I have time for them.
Okay. Thanks, Andy. We do have a couple of questions over here. Let's see. A couple of questions from the Finger Lakes. Can you speak to fungal infections, mold problems, and data on frequency and severity? Large issue in the Finger Lakes.
That's the question, how we're going to look at that issue. So fungal pressure is different, of course, everywhere. And there's a few places on the planet that are really blessed. And one of them is California, one of them is Chile, and one of them is Washington, Eastern Washington. The rest of the world has a lot of problems with maintaining and controlling fungal pressure and fungal diseases. And they work in concert, not singly. In California, we normally have powdery mildew outbreaks, but we don't have downy mildew at the same time. We don't have black rot at the same time. So it's easier to approach those things. But the good news is we have resistance to most of those characteristics. We're probably also going to be using a multiple approach to controlling these things, and a breeding plus minimal spraying programs where you'll spray once in the start of the season and you'll finish the season with a cleanup spray to make sure we're not developing more aggressive strains of fungi that overcome our resistances very rapidly. And again, that's the reason we want these multiple strains of resistance too, not just a single form of resistance, but multiple ones in one background. So it's much more difficult in the Finger Lakes. It's even more difficult in the southeastern United States. We now have the PD-resistant varieties going down there, and it's going to allow them to keep the grapes alive. PD kills the grapevines, remember, but it's not going to prevent black rot and powdery mildew, downy mildew, and anthracnose. That's the bigger issue. So we need to keep working on making those more effective in that regard too.
Okay, I have another question for you, Andy. This is in regards to the CRISPR technology that seems to be really picking up a lot of steam in the ecology and evolutionary biology groups. Are you seeing any promise for that technology in grape breeding? I know most of your work is classical breeding, but I wonder if CRISPR offers any insight into disease resistance.
Yep, very much so. And I think all these new technologies are going to have tremendous potential. We're going to have to get beyond the societal objection to them, to the genetic manipulation, as opposed to things that could be done more naturally, I guess, if you want to look at it that way. It's going to be a process that's going to have probably repercussions beyond what we see now. So it's going to be one of those things that should be approached cautiously. There shouldn't be too much promise emphasized there. Again, working with single genes, not with multiple loci, is going to be easier. And being able to control the way those genes interact and the epigenetic interactions there as well is going to have a bigger role in this than I think as well. But it's another approach that will allow us to move more rapidly. Again, moving grape is tricky because we have to get that final stage where we've changed something, and then we have to look to see how it produces these compounds or allows resistance in the fruit. And then, of course, we have to do that at the wine level as well. So it takes multiple years just to get the material to look at these things oftentimes. But I think it's going to have a role, and I think we'll see that relatively rapidly too.
Great. Well, thanks, Andy, for giving the presentation and for joining us today. Hearing you speak it really is it's a great honor to have you here. Let's see if there are any other questions. Looks like what are the biochemical changes in the vine that result in disease resistance? So how does disease resistance happen?
Yeah, it's actually a good question. And the simpler forms that we understand the most effectively are controlled by single genes. And that's the good news and the bad news. And there's this constant battle between the pests and the pathogens and the host plant in terms of evolving more aggression or evolving more resistance in these situations. So it's a nonstop fight. And the resistances that are more permanent tend to be multigenic. They're controlled by many genes, not by one, and much more complex. But we've got to the point where we know where those are. We call them R genes most of the time. They're in clusters and they form all through the genome. They're in areas that have tremendous incidence of transposons, which are these jumping genes that replicate themselves by moving in and out of the genome. So they evolve very rapidly. And it's not clear whether that's a good thing or a bad thing, but if it could be manipulated in a CRISPR-like situation or scenario, it might be a way to approach more durable resistance in those cases too. Some of those biochemical changes are phenolic. They're based on damaging the pathogen in that sense, and a lot of them are hypersensitive, where the infection occurs, it starts, and the cells kill themselves to prevent them from going any further. So it's a programmed cell death situation, and it also fits into most of these R gene backgrounds too.